Hello, this is my group, which is group 7. Our topic will be metagenome of archaea bacteria in Amazon River. Our group member consist of 4 people, which is my name is Muhammad Ishaq bin Zakir, Nur Awadah bin Muhammad Zahirin, Ivy Adrian Yunus, Nur Ismani binti Idham. Our group choose Thermoplasmata archaea, genocide 582 BP, accession number HQ 287562.
This article is about the archaea in natural and impacted Brazilian environments. Brazil is noted for its natural richness and variety of biomass, making it an appealing sampling location for such investigation. This is the writer or reference. The bioinformatic tool used in this research were the next generation sequencing, which is PCR and qPCR. The use of the different version of the multiple databases also mentioned in the research.
This is the major results obtained in each environment studied based on the article, which is the soil, sediment, and water. By using the certain bioinformatic tools to obtain the results, regardless of whether the samples used in the investigations were water, sediment, or soil, the researcher found that the markers utilized to characterize these species were the 16As, rRNA, mCRA, or AMOA genes. Taken together, the findings show that Euryarchaeota was the phylum most usually detected in water and sediment samples, while Taumarchaeota was more prevalent in soil samples, with most ecological debate centered on methanogenesis and ammonia oxidation.
Our group chose Thermoplasmata archaea.
For analytical method, we use 3 tools to find closest organism and article, which is the NCBI, +N, and cluster omega. This slide shows the summary of how we conduct the search. So let's jump into the videos. Click Amazon River. Choose our database. And then under the genomes, click the nucleotide. Please click the archaea and genomic DNA RNA because we want to focus on that. On the right option, click the uncultured thermoplasmid archaeon. Choose the top of the option. Then click the fast A. As you can see on the title, we can see that we choose the title of article as the main references. Open the +N and pass the fast A that we copy. There's no need to change much, and then click the plus. The data was running. The result was appeared, and we need to choose the closest organism of thermoplasmid. Just choose beside the other name of thermoplasmid. That's the under archaea kingdom. This is not. Alright, this is the one. Click the sequence ID or the gene banks. and then click the FASTA. This phylum is Eukaryote, and then open the cluster omega, change protein to DNA, pass the sequence of thermoplasmid and the eukaryote is FASTA. Or put some space under each of FASTA and then submit. The data we're running. It will take time. Alright, so the result has appeared. This is the alignment between thermoplasmid and eukaryote. The star shows that it has many of significance and similarities. On the result summary, click the percent identity matrix, and they can show that thermoplasmid and eukaryote have same 96%.
There is 4 bioinformatic tools occur in this research. The first one is NCBI, the National Center for Biotechnology Information. Is a comprehensive database of biomedical and biotechnology information that includes a wealth of data related to biological sequences, genetic maps, and literature references. NCBI is one of the most comprehensive and widely used database for searching and retrieving scientific information related to any type of organism, including thermofibrils. The advantage of using NCBI include its extensive data, resources, and ease of use. Next is Google Scholar. Google Scholar is a search engine that indexes academic literature from various sources, and it allows users to search for articles, books, and author material based on the author, title, and publication. The advantages of using Google Scholar include its ease of use and broad scope of coverage, which includes not only scientific publication but also books and conference proceedings. The third bioinformatic tool is BLAST. BLAST is Basic Local Alignment Search Tool. BLAST is a powerful tool for searching and comparing nucleotide or protein sequences. It can be used to identify similar sequences in the database and to obtain information about the sequence, such as its biological source and related organism. The advantage of using BLAST include its speed and accuracy and the ability to analyze large datasets. Last but not least is Cluster Omega. Cluster Omega is a program for multiple sequence alignment and auto comparison of biological sequences. It allows users to align and compare multiply sequences to identify similar regions or patterns. The advantage of using Cluster Omega is its very high accuracy and efficiency and the ability to align a large number of sequences.
Databases have revolutionized the way scientists access and retrieve information and have become essential tools in their research. However, This database also have limitations and potential issues that need to be taken into consideration when using them. The first one is the disadvantages of NCBI. The amount of data on NCBI can be difficult to navigate, particularly for research who are new to the database. The availability of data can vary depending on the taxa or research question of interest. Google Scholar search results can be cluttered and difficult to navigate, and there may be inconsistencies in the publication metadata can affect the reliability of the results. The third one is the blast. The result can sometimes be ambiguous and difficult to interpret, particularly who are not well versed in the subject area. And the last but not least, the cluster omega. It can be difficult to use and it has a limited scope of coverage.